Omega Quant presents a three-part educational series on the basics and the benefits of omega-3 fatty acids, with a special emphasis on the HS omega-3 index blood test. Part 1 of this series covers the basics, Part 2, the HS omega-3 index, Part 3, the benefits of increasing omega-3 intakes, specifically looking at cardiovascular disease, dementia, and mental health. Your presenter is Dr. William Harris. Dr. Harris is an internationally recognized expert in omega-3 fatty acids and heart disease. He's the recipient of 5 NIH grants and has over 120 publications relating to omega-3 fatty acids. Dr. Harris currently serves at Sanford Research Center in Sioux Falls, South Dakota as the Director of Cardiovascular Health Research. He's also serving as a professor at the University of South Dakota Sanford School of Medicine, and Dr. Harris is the founder and president of Omega Quant.
Thank you, John. This is Dr. Harris. I'm going to discuss in this slide presentation, which takes about an hour and 10 minutes all told, 3 different segments about omega-3 fatty acids. I'm going to talk about the basics here, then we'll talk about, as John mentioned, the omega-3 index, and then some of the benefits of eating more omega-3. Within this first section on the basics, we'll first of all discuss the differences between the omega-6 and the omega-3 fatty acids. We'll look at mechanisms of action of the omega-3s and some current and potentially new sources of omega-3 fatty acids. The omega-3 fatty acids and the omega-6 fatty acids together make up the essential fatty acid families for the human diet. Beginning on the left-hand side with the omega-6 fatty acids, we see a linoleic acid is the principal fatty acid in this group in our diet. Structurally, chemically, it's an 18 carbon. This is carbon 1. This is carbon 18. It has 2 double bonds, and it's omega-6 structure. What that means is, if we look at the tail end of the molecule, it has a unique configuration. If we call this the alpha carbon and this the omega carbon, then beginning from the omega carbon and counting 6 positions back, omega minus 6, is where we encounter the first double bond. And this is the same for all omega-6 fatty acids. They all have this same structure at the end. Linoleic acid can be very sparingly but somewhat converted to arachidonic acid, which is probably the most important omega-6 fatty acid metabolically. It's the precursor for many important molecules in the prostaglandin and leukotriene cascade, which control a variety of cellular metabolic steps. Omega-6 fatty acids are plenteous in our diet. They come from a variety of vegetable oils. Corn oil, safflower oil, sunflower, soybean oil has a fair amount of omega-6. So there's not a concern for getting more omega-6, but omega-6 fatty acids are good for us. We do not want to reduce the omega-6 intake, as some people are calling for. On the other side, the omega-3 family, again, the structure here, counting from the omega carbon, is the third double bond. The third carbon has the first double bond. And that structure is not interconvertible. We haven't got the enzymatic systems to convert an omega-3 to an omega-6 or vice versa. And so we have to have both families in the diet. The principal omega-3 fatty acid in plants, in plant oils, is alpha-linolenic acid. It is found in very few plant oils, but in some it's quite prevalent. Particularly flaxseed oil is quite rich in, I'll call it ALA, alpha-linolenic acid. ALA is also in canola oil and soybean oil. Most of what we get in our diet is from soybean oil, since we consume so much soybean oil in America. There is very limited conversion in the body of ALA to the omega-3 fatty acids, the long-chain omega-3s that are found in fish oils. Perhaps less than 5% of ALA is converted to EPA, and less than 0.1% is converted down to DHA. In focusing on the omega-3s in fish oils, we have, again, EPA, which stands for Eicosa pentaenoic acid. Eicosa means 20. It's a 20-carbon molecule. Penta, it's of course 5. It's 5 double bonds. And then enoic acid is just the chemistry term for a fatty acid with multiple double bonds. EPA is what we'll call it in this presentation. DHA is docosah, which is 22 hexa 6 double bonds in again the standard omega-3 configuration here. EPA and DHA are found in oily fish and in fish oil capsules. The best sources probably are something like salmon steaks, which provide a tremendous amount of omega-3 fatty acids, a very good source. We'll now go on to look at some of the cellular mechanisms of action of omega-3, and we're not going to go into great detail on this, but simply to give an overview. One of the most well-known effects of omega-3 fatty acids is the reduction in blood triglyceride levels. And the way that happens is that when you eat omega-3s, the liver reduces its synthesis of triglycerides, and thus it secretes less triglyceride into the blood. Another mechanism by which omega-3 fatty acids affect metabolism is by changing the membrane composition, actually the physical chemical properties of membranes, and we'll see that in just a moment. Finally, the omega-3s can change the balance of pro and anti-inflammatory eicosanoid pathways, which can help change the inflammatory milieu in the body. I'm going to show just one slide that's quite a complex slide, but a very nice one that illustrates all of these factors. And this is a slide kindly provided to me by Drs. Wu and Mozafarian. It's not published yet, but it's a figure in a review article that they're working on. It's the molecular pathways that are affected by omega-3 polyunsaturated fatty acids is the title. And I would note here that sometimes the term N3 is used. It's the same, means the same thing as omega-3, so they can be used interchangeably. The omega-3 fatty acids can have effect on cells outside of the, in the bloodstream actually. These are polymorphonuclear, or white blood cells. This is platelets. Platelets are the cells that get activated when you have a cut, and they initiate the process of clotting. The omega-3s, when they're on board, will reduce the stickiness of platelets and make your blood less likely to clot, which is a good thing to a point. There is no increased risk of bleeding in people taking omega-3s. That's commonly misunderstood, but that is the case from the randomized trials. We know that there's no increased risk for clinically significant bleeding. Within the cell, this now here is the cell membrane, and the cell membrane is a mixture of phospholipids that are on the outside and on the inside, the outer leaflet, the inner leaflet. The omega-3 fatty acids are part of this membrane when we eat them. When we don't, there's very few in there. There's always arachidonic acid in the membrane, and these long-chain important omega-6 and omega-3 fatty acids are sitting in the membrane. They affect the actual fluidity. The way they've stated it here, they changed membrane fluidity, they modified different segments, and they alter protein function. The proteins that are buried in the membrane behave differently if the fluidity of the phospholipids around the membrane is affected. So that's one way we think the omega-3s, just by changing the flexibility of the membrane, it changes the function of many of these proteins. Within the cell, again, the omega-3s can be produce both arachidonic acid and long-chain omega-3, EPA particularly, can be used by cyclooxygenase enzymes, cytochrome P450 enzymes, and lipoxygenase enzymes to produce a variety of what we call active eicosanoids, and that affects metabolism in a variety of ways. Omega-3s can actually affect the DNA, can in fact turn on some genes, turn off some genes, and by virtue of doing that, that's the way it reduces triglyceride levels, by activating certain gene products and proteins that are produced. So the omega-3s have a wide variety of effects in biology, only summarized briefly here in this very nice figure. Let's look at some of the sources of omega-3, EPA and DHA. They come preformed. We get them in fish, and we can get them in dietary supplements. Just a quick list of some of the oilier fish: herring, salmon, whether it's wild or farmed, about the same amount of omega-3. Mackerel is rich. Some oysters, remarkably, have a lot of omega-3 in them. Sardines and albacore tuna, the white tuna, has got more omega-3 than the light tuna. Fish that are not on this table because they have very little omega-3 would be things like catfish and perch and bass. And tilapia, very popular these days, very little omega-3. And so there's a wide variety of omega-3 content in fish. Dietary supplements is another way, of course. Fish body oils are the standard fish oil capsules that you can get at the drugstore or the health food store. These would be typically a 1,000 milligram capsule containing 30 to 50% EPA and DHA. So if you wanted to get about 1 gram of omega-3 from the most inexpensive fish oil pills, which are about 30% EPA and DHA, you would need to take 3 of these pills. Fish oil concentrates are more expensive. They're more concentrated, hence the name, between half a gram and 1 gram of omega-3 per capsule. And this 1 gram omega-3 capsule is actually about a 1.3 gram. It's a bigger pill. To get 1 gram of EPA and DHA into this single pill, the pill has to be bigger. The prescription omega-3 that's available in the United States is called Lovesa from GlaxoSmithKline. It is about 0.85%, or excuse me, 85% EPA and DHA, so 850 milligrams per single capsule. And we'll see several studies coming up that used 1 capsule of Lovesa, or the omega-3 acid ethyl esters, which provided 850 milligrams of EPA DHA. So these are standard sources of omega-3. In the future, there are going to be other sources. And just to go over this rather complex graph quickly, the omega-3s actually start in nature by being produced by microorganisms in the ocean, microalgae. For example, single-celled algae produce the omega-3s from sunlight, using sunlight as energy. And then typically the fish, actually small crustaceans, will eat the microalgae, and fish will eat the crustaceans. The omega-3s get incorporated in fish tissues. Fish don't make any more omega-3 than we do. But fish become a good source of omega-3 for humans. Of course, fish can be caught and the oil rendered to produce a bulk oil, which can be reduced to a dietary supplement, which we would also take. These microalgae can also be used as a source of omega-3 for food, for fish in aquaculture. And the microalgae, the oils can be extracted into bulk oils, which can be made into nutritional supplements, or we can be eaten directly by us. So all these pathways eventually begin with the microalgae. On the other side, genes can be removed from these algae, or from these single-celled organisms, and put into some plant, either crop plants or pasture plants. The best example here is soybean. You can put DNA from other flowering plants and algae into soybeans and produce an omega-3 rich soybean oil. But some of the products of these now genetically modified foods can be either eaten directly or fed to animals to increase the omega-3 content of foods. So these strategies are going to be, are being used now, and some of them will actually come into reality as we see the need for greater sources of omega-3 without actually harvesting all the fish in the ocean. The American Heart Association has recommendations for fish and fatty acid, omega-3 fatty acid intake based on different populations. Patients who have patients without documented coronary heart disease. The AHA recommends eating a variety of fish, preferably oily fish, at least twice a week, and include oils and foods rich in ALA. I'm not sure how important that is, but I would point out that if you actually ate oily fish twice a week, you would get between 400 and 500 milligrams of EPA and DHA per day on average. So that is a reasonable target intake, in my view, for people who want to know how much EPA and DHA they should get each day, 400 to 500 milligrams. Patients with documented coronary disease, people who have known heart disease, are recommended to take a higher amount, about 1 gram of EPA and DHA. It's about twice as much as up here, preferably from fish, but supplements can be used and typically would be used. Patients needing triglyceride lowering, 2 to 4, actually 3 to 4 grams of EPA and DHA per day is a triglyceride lowering dose. Other doses, 1 gram, half a gram, will not lower triglyceride levels. So this is just for a small group of people. So that will end part 1, the discussion of the basics, and we'll